Hello, everybody. It's Bob Vonderheide from Philadelphia, 92 degrees here, as many of you know. And we've got a hot 1 hour for everyone. Welcome to our annual Breakthroughs in Action. And this is a collaboration of Breakthrough Challenge and the Abramson Cancer Center. I'm the director of the Abramson Cancer Center. It's great to be with all of you today. And I just wanted, before we get going, to make a couple of opening remarks. First of all, it's a real pleasure to share the science that Breakthrough Challenge is funding and for you all to meet 6 of the scientists doing the work. The Abramson Cancer Center is really fortunate to be a recipient of the funds from Breakthrough Challenge, and it's really an honor. We look at these funds as really catalytic funds that get us going toward breakthroughs. It doesn't fund incremental science, it funds new ideas. As all of you know, this is the 11th year of Breakthrough Challenge, which has now raised more than $2.5 million, which is absolutely amazing. 100% of participant-raised funds directly supports cancer research here at the Abramson Cancer Center, and we are very, very grateful. You know, over the years, we've now seen the countless opportunities that this funding has provided for our faculty, our patients, their families, and really bringing together leaders and community members here in the Philadelphia area all toward this great cause. We are particularly grateful to Chuck Zent and the Zent Family Foundation for their ongoing, continued, and vital support of Breakthrough Challenge, in particular, the annual Match Challenge campaign, which is again active this year, really helping us to raise even more dollars for cancer research here at the Abramson Cancer Center. So thank you, Chuck, and your whole family. Look forward maybe to seeing you in September. So over the 11 years, we've now been able to fund 58 faculty members who've received grants from the Breakthrough Challenge. We call them Richard Buzz Cooper Scholars. Buzz Cooper was a Abramson Cancer Center director a few before me. And you'll hear from 6 of these individuals today with short presentations and an opportunity to ask questions. Before we dig into that, I just want to remind everybody that this year the event will be Sunday, September 15th. So make sure that's marked on your calendar. That's the date of the in-person bike ride at the Daniel Boone Homestead, the same location that we've been at for the past several years. And then in addition, the week before, there'll be a virtual event for spinners, runners, walkers, cyclists, and that begins on September 9th, and we can get you all that information, how to set up your team, how to register, how to participate in any number of ways. So it's always a great event for the Abramson Cancer Center that day in September, and we look forward to that very, very much. What is that event all about? It's about fueling science and breakthrough work. And so today, what we'll do is we'll hear short presentations from 6 presenters, 6 faculty members who are Buzz Cooper Scholars. I may ask them a question or two, but we'll have time at the end to take your questions through the chat. And Carolyn Brown is online helping me monitor the chats. Okay. So without further ado, our first presenter is Dr. Zev Binder. Dr. Binder is assistant professor of neurosurgery and is working on an exciting project relevant to glioblastoma multiforme. Zev?
Thank you very much, Bob. Let's see. There I am. And all right, and we have the screen up.
Okay. Looks good.
All right, let's go ahead and. Okay, all right. So thank you very much, Bob, for inviting me here to talk, and thank you also everyone on the call for all the work that you do for generating funds that has allowed me to actually drive this work forward. So I'm going to go ahead and present on my project, which is development of an autologous humanized mouse model for the study of immunotherapy in GBM. Or really, is there a way for us to incorporate all the elements of a model system to come from the same source? So I work on glioblastoma, or GBM. So GBM is the most common and lethal primary brain cancer found in adults. And one of the driving forces of that lethality is that it's an incredibly invasive tumor. So you can see here that there's a rather large tumor bulk, but if you look at these autopsy slices, all of these black dots here represent individual nests of tumor cells. And so even though there's a bulk of a tumor, individual cells sort of spread out through the rest of the brain and are able to drive recurrence and growth of this tumor. And what that means is that non-targeted approaches such as surgery or radiotherapy or chemotherapy are really considered non-curative because they're not able to go out and find these isolated nests of tumor cells and eradicate all of it. So the current median overall survival for GBM is under 2 years. So this is a disease that is in desperate need of some new approaches and new interventions. So what is it that we at the Abramson Cancer Center are trying to do in order to address this and really account for this invasive nature? And my group, along with the clinicians, has really chosen immunotherapy as our approach. We feel like this really offers the tools and opportunities. Oops, it's missing one of the slides there, I apologize. It really gives us the opportunity to find those isolated nests of tumor cells. So over the course of the last 7 to 10 years, we've had the opportunity to conduct several clinical trials. We've completed 2 and are in the midst of our 3rd right now, using chimeric antigen receptor T cells, or CAR T cells. So CAR T cells are when we take the patient's T cells out of the body and we re-engineer them, or we educate them to target something that we know is on the tumor, bring them back, introduce them back into the patient, where they're able to go through the patient's body and actually find those tumor cells and kill them. And it's an approach that we really strongly feel is going to have a significant chance of success in combating this kind of tumor. So my lab does a lot of work focused on the development of these therapies. And our standard approach in the lab when we're working with animal studies is to take a mouse with no immune system so that we can introduce human elements to it. And we put in the human tumor, and then we take T cells from a healthy donor, make our CAR T cells, and administer those to the mouse. The problem is that with an immunodeficient mouse, you are missing that inherent immune system. And we know from our clinical trials that that immune system plays an incredibly important role both in its interactions with the tumor as well as with the treatment itself. So we're missing a really important element in this model system. So the standard way that people try and address this is actually inject them into the mouse, give it some time, and that mouse will then begin to develop actually a human immune system. And so what people do is they get a bone marrow sample from a healthy donor. They get the tumor model, tumor cell line from a patient. And then again, from a healthy donor, you get your peripheral blood to isolate your CAR T cells. But the problem is you have 3 separate sources of the elements going into your model system, 3 different people. And yes, they're all the same species, but there are differences between them. And so you're going to end up with interactions between these 3 different elements of the model system that is not reflective of what actually happens in a patient where all of these elements come from the same person. So my project has looked at, can we obtain these 3 elements from the same source, i.e. the patient, and will it work in creating a model system that will hopefully give us higher fidelity results in our preclinical work so that we can have more faith when we take it into the clinic that we'll get good results. So we take advantage of the fact that the patients go under anesthesia for their surgical resection, and of course with consent, we do a bone marrow biopsy while they're here for their surgery, and that allows us to get our bone marrow stem cells. At the same time, we take a piece of tissue from the surgical resection, and we use that to establish our tumor model. We use one called a GBM organoid, or a GBO. And then we get a peripheral blood draw, which isolates T cells and allows us to create our CAR T cells. So we're getting all 3 elements coming from the exact same patient. And really the first question was, is this even going to work? GBM patients are known to have pretty crappy immune systems at the time of diagnosis. Not going to go into the rationale behind that, but there was really a big question of whether or not we could get good enough quality material to humanize the mice. And you can see here, this is looking at the percent of immune cells in the mouse that are human in origin. This was from a healthy donor, and this is from patients. And you can see that while they are lower from the patient, we are still able to get human immune cells in these mice using patient material. So we at least answered that first question successfully. The next question was, are they just going to stay in circulation, or are you getting these immune cells to behave the way they do in a patient, which is that they infiltrate into the tumor? And so this is a slice of the mouse brain. And we can see here, these are 2 locations that actually have tumor to increase cell density, but really this is the pathologist affirming this for me. I didn't see it on my own. And then I went ahead and stained these same pieces of tissue with a marker that is specific for human immune cells. And you can see all of this dark brown here are positive human immune cells in the same vicinity where there was tumor. So we are able to humanize the mice. We are able to then get infiltration of that immune system into the tumor. And what we're working on now is trying to figure out what elements of this immune system are actually nicely reproducing what we see in the patient. I don't think every different cell type is going to be there and completely functional, but if we can figure out what elements are doing a good job at modeling it, those are the elements that we're going to focus on as we continue the research. And so with that, sticking in under my 6 minutes, I wanted to thank again everyone here for helping with the contribution, as well as all the lab members and associated groups that have helped me on this work. And I did, just in sort of a self-plug, Bob mentioned that this is sort of the generating spark, and this funding has allowed me to generate the preliminary data that has then allowed me to go on and get some NIH funding for this. So I have funding carrying on to continue this project.
Yeah, thanks, Zev. It's really exciting. Give us a sense if this new model will help you find new therapies faster or more accurately, or both.
So it's going to be more accurately. I don't think, unfortunately, it's going to be speeding it up, but it's more that we cure mice every day in lab of GBMs, and that doesn't carry over to the clinic. And so I'm really trying to help bridge that divide between what we see in the lab and what we see in the clinic.
Yeah, it's going to increase our batting average.
Yes.
Okay, Zev, stay tuned. We're going to come back to you near the end of the hour. We may get into that return on investment point you made. So we'll turn to our next speaker, who is Linda Jacobs. Linda is clinical professor of nursing and has been funded on a survivorship project and is here to tell you all about it. Linda?
Okay, so I want to thank you for inviting me to present. I'd like to thank the board for the funding that's allowed my team to conduct the study. We used text. Hang on, let me move this out of the way. We used a text message to increase the risk-based survivorship care among survivors of childhood and young adult cancer survivors. And that's because approximately 85% of pediatric and young adult cancer patients now survive greater than at least 5 years or greater, and many of them are lost to follow-up. And there's lots of reasons for that, because these are young adults and they want to move on with their lives. So what is
Can we
PENNY?
ask you to unmute your screen?
I'm sorry, are you talking to me?
Yes, please, you're muted, your video.
Oh, hang on a second. I didn't think I was. I can't get
Victor, I can see it.
my screen. You mean the slides are muted?
Your slides are fine. It's your video you can't, but go on.
Okay. So the agent that we use, the chatbot, was named Penny, and it's a conversational agent. The text messages, it's bi-directional, and it is communication and motivational cues to promote adherence among these patients who enroll. Penny has been designed to schedule follow-up appointments, lab scans, and specialty appointments. It also monitors compliance with the appointments. It's a 2-arm randomized controlled trial and explores how these multi-level factors impact acceptability and effectiveness of the strategy. We also collect qualitative and quantitative data. The study opened on September 11th, 2023, and we utilized the electronic phenotyping core at Penn at the Abramson Cancer Center for our sample. Our inclusion criteria included patients who were diagnosed between birth and 39 years of age, and had had no follow-up care for at least 3 years. The age range of these patients is quite wide, 25 to 77, although most of the patients were under 45. We screened 239 patients, and 110 were eligible, with 129 ineligible. This is interesting in that 98 of the 239 patients were receiving care elsewhere, which was somewhat surprising, because in many of the studies that look at this population say that they're lost to follow-up, when in fact they may be lost to that institution, but they're not lost, they're getting follow-up care. And 71 out of 110 were in fact lost to follow-up. So 39 patients out of 110 agreed to the study, but only 28 consented. 15 were randomized and consented to Penny. 5 actually completed their appointments. 3 have appointments pending. 1 was no-show, and 6 did not schedule yet. There were 13 patients randomized to usual care. 4 scheduled and completed appointments, and 9 did not schedule. However, well, as you can see, there's no significant difference between groups. And why is that? That's because we surmised, and we actually had done this with another study, that these patients Just the act of contacting them reminds them that they need to have an appointment. So patients who were not making appointments, who were in the usual care group, actually wound up making them because someone had contacted them. But that's okay. So one thing we learned from our survey was that patients in Penny had good understanding of the need for care. They had high confidence in their knowledge of the care needed. And however, prioritizing follow-up care was low in this group. And if you can guess that this is a population that has lots of things going on in their lives. They're in college, they're just getting married, they have small children, so they generally do not follow up. So we got a lot of quotes from our patients, and a few of them, you can see what they are here. Patients really felt that getting text messages about healthcare really did help them and made things more convenient and easier for them. And I'll just add that yesterday I got a call from one of the patients who's enrolled in the project looking for some information because he was diagnosed with a new cancer. I haven't seen him in 10 years, but he actually was getting in touch with us and wanted us to help him make his appointments for surgery, etc. So our next steps, we submitted an amendment to the IRB June 1st, and it's been approved. We again are going to gather our population from the electronic phenotyping core. We're going to open the study September 1st, or continue the study, I should say, with a few changes. Patients will be diagnosed from birth till 39 years also, but they've not been seen for care in 2 or more years. We found that the majority of the patients who hadn't been seen, there were many patients who hadn't been seen in 2 or more years, and they did not make appointments. The age range of the patients, again, will be the same. And we'll, instead of contacting groups of 20, which we were doing, we're going to send a text blast to 264 patients, which are the ones that we identified through the phenotyping core. We will not be contacting patients with phone calls, and we're not going to be randomizing them to one group or the other. And all the patients in the group will be getting surveys. So it'll be a little bit different. And some of these changes were recommended by our vendor, which is a company that manages the chatbot, and they thought that this might improve our our enrollment, and it actually will make it more real life also. So that's all I have, and I'll look forward to questions at the end, as well as from Bob now.
Yeah, thanks, Linda. I think we are really leading this type of using this type of communication with our patients. Is it spreading across the country to other cancer centers now following this example?
There has been a number of chatbot studies, not necessarily encouraging patients to come back, but doing other things, questionnaires, etc. This is a population that, if you were familiar with the literature, you'd see it's considered underserved and neglected. Also, there has not been great inroads in managing them and dealing with them post-treatment in terms of what actually happens to them. Many of these patients develop second cancers or other very significant medical issues as related to the cancer treatment they received. I see many of those patients, and it's pretty surprising. So it's a good population to study.
Super important population who does a lot of texting. So this is
yes.
Okay, thanks, Linda. Stay tuned. We'll come back to you. If you can not de-share so that Yannis can share, I'll introduce Yannis Virginatis, who is here, assistant professor in radiation oncology, and he has a project that involves a new technology called flash proton radiotherapy.
Yeah. Thanks, Bob. Hi, everyone. Can you see my screen?
Yes, it looks good.
Okay, that's great. So first of all, thank you for the opportunity to present our project here, and also for the funding support, which allowed me to generate data. You'll see we have a publication from this work, and also allowed me to submit a bigger grant to NIH, and in a few months to Department of Defense. So my project is about increasing the therapeutic window in cancer treatment with flash proton radiotherapy. So we know that radiotherapy is one of the major treatment modalities against cancer, and more than 50% of cancer patients that receive radiotherapy as a monotherapy, but also in combination with chemo and immunotherapy. And we know that every cancer treatment has causes like normal tissue toxicity, and the same applies for radiotherapy. So despite the technological advancements in the past decades, we still cannot avoid the normal tissue toxicity during the radiotherapy, and we mostly talk about the long-term effects that develop years after radiotherapy. So we are really urged to find new technologies that will allow us to protect or minimize the normal tissue toxicity, but at the same time control the tumor growth. So one of these technologies, which is exciting technology in the field of radiotherapy, is called flash radiotherapy. And what that really means is that we deliver exactly the same dose as the conventional or standard radiotherapy that we currently use in the clinic, but in a very, very short period of time. And what has been shown so far about the flash radiotherapy is that spares normal tissue. It means that causes less normal tissue toxicity, but at the same time, it keeps exactly the same efficacy on tumor control as the standard radiotherapy. And just to give you an example about the time scale and the difference between the conventional and the flash radiotherapy, you see here in the red bar graph that conventional radiotherapy now in the clinic takes minutes in order to treat a patient, several minutes, compared to the flash radiotherapy, which it's several lower magnitudes of minutes. In order to deliver flash radiotherapy, it takes a few milliseconds or 100 milliseconds. So the flash actually term is not a new term. So it was started back in the 60s, where they have developed some technologies, and they were able to deliver ultra fast dose rate delivery of the radiation, mostly on cells. And they saw that with flash radiotherapy, they can cause lower toxicities in cells, but in some tissues. But due to lack of technology, they stopped all the experimental procedures. So there was a paper from two groups in France and Switzerland in 2014 that they reinvigorated the whole flash field and showed that flash can spare the lung toxicities in murine models. And in 2019, our group, Radiation Oncology Department, we released the first paper showing that flash proton radiotherapy, because the first paper in 2014 were talking about electron radiotherapy, which is not very common in the clinic. So we released the first paper in 2019 showing that, again, the flash proton radiotherapy can spare the normal tissue, intestine, with the first experiment performed in intestinal tissue. And actually, this is one of the most highly cited papers in this journal. So in 2021, we have the first clinical trial at Cincinnati Children's Hospital, and they performed the flash versus standard radiotherapy, but that was a clinical trial for efficacy only to see if they can deliver flash radiotherapy on patients. And during this time period, we published actually 5 high-impact research articles, including the one that I'm currently investigating. And we hope that in 2025, we're going to initiate a clinical trial in our department for head and neck cancer patients. So actually, I'm thrilled that this project got published a few months ago, and of course, the funding support were acknowledged in this paper. And in this project, we are trying to address a very big issue in the clinic. And this is when patients treated with radiotherapy, especially for breast, lung cancer, head and neck, even pancreatic cancer, they get incidental radiation to the heart. So years later, they develop fibrosis, which is the deposition of collagen in the tissue, the radiated tissue, and they perform lowering the cardiac regulation. So they have cardiac toxicities. And this actually is very common in pediatric tumors, like when the kids are treated with radiation. 20 years later, they are admitted to the clinic with severe cardiac toxicities. So we're trying to assess if flash radiotherapy can mitigate these long-term effects, toxicities. So this is just a recap quickly what we performed in this project. This is the model that we use, the mouse model. And as you can see here, we have the ability to deliver precisely on the bottom of the heart, the apex, because it's the one most commonly irradiated site in the clinic. And we can deliver, and we know exactly how much dose we deliver because we have the ability to perform the treatment plan. And this is a recap of the whole study, different types of analysis. I'm not going over this one, but just to show that it takes also 30 weeks in order to have the long-term effects, in order to mimic the clinical setup that takes years to develop the cardiac toxicities. And just to mention that we also perform two-dimensional echocardiography exactly as we assess the clinical cardiac performance. So one of the key points that we found in this project is the expression of this marker, the TGF-beta1, which stands for transforming growth factor beta-1. And why this is important, this is a key molecule to initiate and progress the fibrosis. And when we collected tissues, cardiac tissue from untreated, which is the very far left, and compare the standard and flash irradiated hearts, you see that this factor is highly upregulated in the standard hearts, irradiated hearts, compared to the very, very low expression level in the flash, meaning that the fibrotic tissue in the standard is in high levels and also affects the cardiac functionality. And as we can see here in the echocardiography, that the distance in the left ventricle is reduced because of the fibrotic tissue, compared to the flash-treated mice that resemble us untreated. That's amazing, and we it was surprising for us. So far, what we have shown with flash proton radiotherapy is that reduce the normal tissue toxicity, and we have performed several models in the cardiac tissue, skin, intestine, and now we also perform liver toxicities, but also compare or in different tumor models. But what is really important for the clinic is that we minimize the motion effect, because now imagine that we deliver the dose in a few milliseconds compared to many seconds or even several minutes. So we don't now care about the motion effect that lung and breast cancer patients has to face. The lower normal tissue toxicity will allow us to deliver higher doses to the tumor. So now we're going to have a better tumor control, not only the primary tumor, but also in the metastatic sites, which is the most important treatment, like for the cancer treatment. And we believe that flash proton radiotherapy is going to revolutionize the whole radiation field, because no one has done that before, and we believe that's going to enhance the biological therapeutic index, so we can treat better the patients, we can have a better tumor control, and most important, we can improve the quality of the cancer patient survivors. So with that, I would like to thank everyone, and have to take any questions to the very end and from Bob.
Thanks, Giannis. Maybe not a question for me right now, but just to make the point that we're able to do flash because we're leveraging our proton technology. You can do flash from electrons, you can do flash from photons, but it's just not really feasible. Protons changes the whole dynamic, right?
Yeah, exactly. Yeah, I agree with that. And protons are very unique, have unique characteristics, because they can deliver the highest of the dose that we are trying to treat for the tumor, but at the same time they minimize the entrance, as we call, or the exit dose of the target, so minimize the normal tissue toxicities. And at the same time, on top of that, imagine that you apply the flash radiotherapy, so we have actually something really powerful tool in our hands.
For sure. Okay, thanks so much. We'll keep going. Our next presenter is Bobby Bowman, who is an assistant professor in cancer biology and an expert on leukemia, and has a funded project that he'll tell us about.
All right, thanks so much, Bob. So I'm going to tell you today about our work looking into cancer evolution, and specifically we're going to be looking into single cell studies to understand how cells change over time, particularly in their methylation patterns. So we do this in the study of acute myeloid leukemia. Taking a step back, when we think about cancer evolution, we want to kind of take a look at where mutations are in the body, and we kind of grow up with the textbook knowledge that all of our cells in our body have the same set of genes and the same set of characteristics that make us unique from one another. About 10 to 15 years ago, we started making seminal discoveries in the field of a growing field called clonal hematopoiesis. And that is that not all of our cells have the same DNA or the same set of sequences, but in fact, we can find small mutations in the blood of totally healthy people. And this is a pretty ubiquitous thing. Anyone over the age of 65, about 1 in 10 will have these mutations in the blood. Now, that does not mean cancer, but that does mean there is some sort of process that's leading for DNA to be mutated. Now, blood is a pretty easy tissue to sample compared to other tissues in the body. And over time, we've started to find these mutations in more and more tissues. And it's not just your blood for a particular set of mutations. You can actually find them in T cells and B cells and precursors to red blood cells. And these have effects on disease biology and can affect things like inflammation or response to arthritis or other inflammatory like diseases. And some of these mutations can also lead to leukemia. And over time, we found that this is not true just the blood system, it's true for many tissues in the body. So understanding how we go from having mutations to actual disease is something that my lab's very interested in. And it's not just the picture of normal human health, it's kind of a picture of how things change through leukemic development. So you can imagine a scenario where we have a whole set of cells. When this clonal hematopoiesis, the initiating event happens, we pick up a mutation in the cell, and those cells start to grow and outcompete other cells. And the way we think about leukemia development is that these cells will continue to acquire more mutations to the point where now they have this cell may develop into a very aggressive growing, fast growing cancer. At this point, if we were to administer leukemic therapy, we might bottleneck this population of cells and select for different clones that may outgrow. So this is the entire process we're trying to understand. In this particular project, we wanted to understand how the DNA methylation changes along this process. So DNA methylation is an interesting facet of biology, and it's a way that we can actually compact and organize our DNA. At the highest level, our DNA is organized into chromosomes and these large structures that are wrapped, where the DNA is wrapped around these protein cores called histones. And the kind of final step of modification is the small chemical alteration leading to a change in a base or a piece of DNA that gets methylated. And the critical enzyme that's important for this methylation is a gene called DNMT3A. And this happens to be the most commonly mutated gene in clonal hematopoiesis. So many people are walking around with mutations in DNMT3A. The second most commonly mutated gene is TET2, a mark that actually removes this methylation mark. So understanding how DNA methylation influences gene expression, influences how cells grow, is really critical to understanding, we think, these early stages of clonal hematopoiesis. On top of that, chemotherapies actually inhibit DNA methylation, are effective forms of therapy. So we are really stuck with this picture of trying to understand how DNA methylation changes with these different mutations and how it happens on the single cell level. So to add a further wrinkle to this, despite DNMT3A mutations decreasing DNA methylation and TET2 mutations increasing DNA methylation, we find that there are a subset of patients that actually have both of those mutations in the same leukemia. And that's a little confusing. And you might ask, like, well, what happens when you have both these mutations? Do you end up with a whole different methylation landscape, or do these things average out? And in general, we see that if you have both these mutations, the methylation averages out. You lose regions of the genome that were once hypermethylated, they now go back to normal. And you lose regions of the genome that were once hypomethylated, and they go back to normal. We are left with this problem, though. If DNMT3 mutations lead to low methylation, TET2 lead to high, and you're co-mutated, is this really a scenario where you have a bunch of cells that have no alterations of DNA methylation? Or is it a scenario where that some cells are high and some cells are low? And this is important for us to figure out, because when we start applying therapies that block DNA methylation, our question is going to be, are we selecting for another outgrowth of cells by that therapy, or are we going to totally change the biology of these cells? So this is what this project's been focusing on, is trying to understand on a single cell level, where are the mutations, where is the DNA methylation, when we apply therapy, what's likely to happen? Our first question is, if we have these DNMT3A and TET2 mutations in the same leukemia, do they actually even co-occur in the same cell, or are they in opposite cells? And our studies indicate, it's a nice little circle plot here, that not only do they occur in the same cell, but these red lines indicate that they occur in the largest fraction of leukemic cells within a patient. So this is a bit of a biology paradox we're trying to figure out, and understanding how these enzymes with opposing functions can associate with a cellular outcome. So we think if we can kind of get an idea of how the methylation is being altered, this will give us an idea of how we can target both late-stage leukemia that harbor these mutations, as well as potentially blocking clonal hematopoiesis expansion. And that's going to have a large role, not only in just the rate of leukemia development, but also a positive effect on human health, as clonal hematopoiesis is associated with much more than just leukemia development, including cardiovascular disease. So that's the kind of snippet I have to share with you today. We've had a great crew of people working on this in the lab, as well as some wonderful collaborators here at Penn and at Thomas Jefferson across the way. So thank you for your time and attention.
Thanks, Bobby. Say a little bit more about how from the clinician's point of view, the value that they're going to get from these new understandings?
Yeah, so these mutations early on, the TET2 medium 3As, we use these mutations to track how well we're doing clinically on regressing a leukemia. So later mutations, after a patient's been on chemotherapy, we will see that they disappear during remission, but these early mutations do not. And they are not good biomarkers of response to therapy. We are hoping, though, that if we can understand how these mutations affect DNA methylation, that those will be better biomarkers of response. And maybe that we're seeing specific changes in leukemia that are being repressed at remission. That may give us a better idea of assessing measurable residual disease in a patient's likelihood to stay in remission.
Got it. Good. Okay, thanks, Bobby. We'll move on, and we'll have Wenbao Yu present. Wenbao is an assistant professor in pediatrics. You're noticing that all the scholars are coming from different departments across campus. It's multidisciplinary. And Wenbao is going to talk about something called plasticity in cancer cells. We can see your screen. There you go. Okay. You're on.
Sorry.
Now we can hear you.
Unmute myself. Thank you, Bob, for the introduction and the opportunity to present my work here. And I'm going to present my funding project, which is aimed to developing computational tool for understanding cancer cell plasticity. In normal cell development, progenitor stem cell, they give rise to different types of cells with distinct functions through various immature intermediate cell states. But in cancer, the cell can be, this process are disrupted, and the cancer can be stuck in a particular developmental branch, or they can reverse back to an intermediate state, or they can switch to another developmental branch. So collectively, this is what's called cancer cell plasticity, is the ability of cancer cells to switch in their identity to involve and resist treatment. So they, in the molecular level, there has to be some certain gene programs be dysregulated. Essentially, gene expression is regulated by a particular type of proteins called transcription factors through binding to a specific type of DNA sequence we call enhancers. Systematically, this regulatory relationship can be depicted by a transcriptional regulatory network. Essentially, the network, the loads in a network are transcription factor or genes. There's a link between them if there's an enhancer between them. So actually, constructing TRNs is critical to understand cancer cell plasticity, but it remains challenging because the complexity of this process and also the requirement of simultaneous measurement of multiple type of molecules from the same cells. So to address this challenge, we propose to develop a computational toolkit for the construction of the TRN by leveraging the state-of-the-art single-cell multiome technology and machine learning methods. So with the method we developed, it's called SimpleTRN, which takes input of experimental measurement of mRNA gene expression and DNA accessibility from the same cells, and outputted cell-specific network, TRN networks. There are two tasks in SimpleTRN. First, to infer the gene and enhancer links, and also the second is to construct the cell type, cell-specific TRNs. We are currently focused on the first task, and through a benchmarking study, we found a prototype of SimpleTRN has improved the accuracy of predicting long gene enhancer links by 30% compared to conventional methods. So we go ahead to applying this prototype to study the high-risk neuroblastoma. So high-risk neuroblastoma is a devastating solid pediatric tumor arising from the peripheral nervous system, and it counts of about 10 to 15% of pediatric cancer deaths, and the outcome hasn't been improved for decades. So we identified 6 distinct cancer states with a profound shift after therapy, indicating there's some transition among these different cancer states. And we also found multiple enhancers and transcription factors that collaboratively regulate cancer state-specific gene programs. And for example, this is at gene EZH2 region, where we found 3 novel enhancer for this gene. EZH2 is a master regulator and an emerging therapeutic target for neuroblastoma. And the enhancer we found has higher accessibility in the proliferating cancer state, which is the most devastating state for this type of cancer. But also we noted that this enhancer also accessible in several other cell states. So these permissive enhancers may indicate the cancer states, enable the cancer state shifts and make the treatment very hard. So we will further investigate this cancer through the progress of the second task of developing SIMTRN. And yeah, with that, I would thank all the breakthrough challenger board and the donors and all the collaborators, my mentors, and thank you.
Thanks, Wenbao. You know, this is, you know, an example of what we call data science and computational science. And we're understanding the cancer cell now at the level of the state, right? It's really getting complex. Just to make the point, you're looking at neuroblastoma, but this approach could be applied to multiple cancers, really any cancer, perhaps.
Yeah. This the method itself can it's very can be applied in multiple. Actually, we already did to applying this to like glioblastoma to investigate the plasticity and the network. Yeah, that's
That's great. That's very exciting. And so thank you for the presentation. We have one more presentation from Dr. Dev Basu. Dev, are you there? Yes, I see you. Dev is an associate professor of otolaryngology and an expert on head and neck cancer, and will talk to us about cancers of the head and neck related to human papillomavirus.
Thank you, Bob, and members of this audience for your generous support. I'm a head and neck cancer surgeon and cancer biologist, and I want to share with you how we've been using some of your resources to help patients with cancers of the throat that are caused by HPV, the human papillomavirus. And most people are aware that HPV is the main cause of cervical cancer, and screening and now vaccination is making a big difference for that disease. I think overall there's less awareness that HPV is a major cause now of head and neck cancer, and these tumors develop from the side of the throat, very often in the tonsil or from the back of the tongue, which is particularly susceptible to this disease. The incidence of these cancers has grown rapidly over the last few decades, and in the US and at least across much of the developed world, they're the most common HPV-related cancer, more common than cervical cancer, and there's no effective screening for them, and they're projected to continue increasing in incidence for some time. These cancers typically respond to treatment using various combinations of chemotherapy, high doses of radiation, and surgery, but that treatment is fairly difficult to endure, and it leaves behind lasting disabilities in patients, often folks who are in the prime of their lives in their 40s, 50s, and 60s. And many of these patients are cured, and we want to treat them less aggressively and leave them with less disability. But some patients do recur despite the most aggressive therapy, and it's hard to tell who those high-risk people are up front. So to meet this challenge, we need new molecular strategies to separate patients based on differences in the biology of their tumors, and which we're just starting to unravel. And ultimately, we need to carefully tailor our treatments to people with the high risk and the low risk biology. we're starting to get some insight into what makes some of these cancers behave more aggressively than others. What we've learned from examining tumors for many patients treated at Penn over the past decade and a half is that the growth of the typical ones is somewhat limited by damage that the virus itself, the viral proteins themselves, cause to the tumor cells. And specifically, the virus pushes tumor cells to divide too hard before they're fully done replicating their DNA. And so when you treat with radiation and toxic drugs, that makes the DNA in these tumors easily fragment to the point of killing the tumor. These DNA fragments also create a signal that activates the body's immune system against the tumor, which also makes them easier to cure. So what we're learning in the patients who tend to have bad outcomes, lethal outcomes from this disease, is that they are much better at repairing their DNA by various mechanisms. And doing so makes it easier for these tumors to grow and to spread to other parts of the body. The stability of their DNA makes these tumors much less visible to the immune system. Also, they don't cause as much inflammation. And so we're trying to use these properties to distinguish the high-risk patients and the low-risk patients front and to treat them differently. And so I don't have much time to get into the details, but what I want to convince you is that we've put together an excellent team of faculty with complementary skills who are working together to tackle this challenge under a Translational Center of Excellence in this disease at Penn. I myself have been working on identifying the features of tumors that do badly and the mechanisms that make them more resistant to treatment. Eric Brown is a real DNA damage expert and cancer biologist who's helped to develop new drugs that target the pathways used by these tumors to resist therapy and prevent and repair their DNA. Ahmed Diab is a new cancer biologist at Penn who's becoming a close colleague, and he's working to understand how we can use treatments that cause DNA damage in these tumors to better activate the immune system against them. Jianxin Yu is a virologist and a real expert on HPV who's developing a new lipid nanoparticle-based approach to reverse the virus's own abilities to shut down immune responses against it. Lovis Sun is a head and neck medical oncologist who's working to bring one of Eric's new drugs into the clinic for the first time against HPV-positive cancer patients who failed standard treatment. And lastly, Nick Lukens is a head and neck radiation oncologist who's working with one of Eric's drugs that's a bit further along in development, this ATR inhibitor, combining it with low doses of radiation and an immunotherapy drug before patients start standard therapy, specifically in patients we know have a high risk of failing treatment up front. So I'll end there. I'm sorry that went by so fast, but I'd be interested in talking to anybody offline who has a particular interest in this disease and our work.
That's great. Thanks. Thanks, Dev. Maybe you can take your screen down and we'll open it up now for questions. I'll start with you, Dev. So this is an incredible team you're putting together, such a multidisciplinary approach on an important disease. If you're successful, what will treatment, how will it look different for patients in a few years' time? What's your hope for that?
So the hope is that we know that a good number of these patients, their tumors fall over with minimal treatment, a very low dose of radiation, limited extent of surgery, no drugs, limited lasting side effects. And that can be the majority of people with this disease, a totally different landscape from what they experience now from treatment toxicity. And then we were able to do that because we can very successfully pick out the people up front who are at risk of coming back in the coming months and years with distant metastases in their lungs and their bones. And we can take a completely different approach because a bit more toxicity is tolerable and acceptable.
Yeah, it's an interesting, this is all about precision medicine, treating the patient for who they are, not the label given to them by the pathology of the tumor. And sometimes we're learning here where we actually have great therapies for patients and we're over-treating sometimes and under-treating folks who need more. So this is pretty exciting. We'll welcome all the investigators to answer any questions, and the audience can put those in the chat. There are already a few that I will start. Some are more general about this program with Breakthrough Challenge. So, and then we'll welcome Bo in a few minutes for some concluding remarks. Does funding from Breakthrough Challenge provide seed funding, and are the researchers able to get additional funding from national grants? So this is the brilliance of this program. So these grants are relatively speaking small, but they get a group of investigators or an investigator going to make the initial observations, and with that data, you can now apply to, say, for example, the NIH for a much larger grant, which might be over $1 million. So we track that very carefully and can say that, you know, every dollar from Breakthrough Challenge turns into $9 from some other organization, and that's really the power. On the other hand, if you just apply straight away to the federal government, the chance of getting that grant is less than 10%. And so by having the funding right away, so our folks can get going, we increase their chances to get the big grants quite a bit. And that is, if you want to call that seed, I guess that is a way of looking at it, but that is what it's all about. Someone asked, are there research projects that were very close to being selected and funded beyond the ones presented today? And how much more money would have been needed to fund the additional projects? So it varies year to year. This year we received 28 applications and we funded 7, which, although that's higher than the funding rate at the NIH that I just quoted, which would have been 2 or so, we did receive many more grants that were meritorious and we didn't have the funding. If we had twice as much funding, we would fund twice as many grants because all the money comes in and goes straight to research. So, and again, then the more seeds that we put out there, the more they grow into much larger grants. And so that's why the work that you all do is so important. The more that's raised, the more we can fund. Absolutely, for sure. I might turn this question and see if I can get some volunteers from our experts that you just heard from. One of the folks says, can you paint a picture of the next 10 years of cancer advancements and where would you hope to be? So maybe I'll ask any of our investigators, they can open their screen and offer a comment on that. Where do you want to be in 10 years? Bobby?
Yeah, I can give a perspective of how we think about this, and really in heme malignancies is one spot, and in many ways, just having access to samples, heme malignancies have really pushed forward a lot of therapies that find their way to the solid tumor. So I think it's still largely about biomarker discovery and which patient's going to respond to which therapy. Specifically in acute myeloid leukemia, this is probably a collection of 8 or 9 different diseases, right? And having the biomarkers to assess which patients will respond to which of our therapies and which ones won't. And like the comment earlier is, are we over-treating patients is an important thing to know. So that's where we are, is certainly trying to understand who's going to respond to what, and trying to figure out which of these patients, if any, will also respond to immunotherapy, which may at times have a broader use case once you've kind of cracked the potential code of what that case might be.
Yannis, you want to weigh in?
Yeah, just mention in terms of radiotherapy, as I mentioned, FLAS is something amazing that we haven't seen that before in terms of normal tissue toxicities. And as I mentioned, just like to recap 2 things. First of all, we're going to be able to deliver higher doses to the tumor, so we're going to be able to control tumor growth in both primary and metastatic sites. But as I mentioned, that even tumors that so far are can be treated with radiation, for example, type 1, like stage 1 and 2 breast cancer that are almost cured, we still minimize the normal tissue toxicity. For example, patients, they live for years and years, but they suffer from dermatitis, skin toxicity, lymphedema. So imagine that we won't have any issues, or at least we have minimal issues on side effects. And imagine that we improve a lot of the quality of life of these patients. So I think that's going to be implemented in the next years in the clinic, and it's going to have a huge impact.
Zev, we'll let you have the final word. You study brain tumors, so
yes, I'll be brief. I think good. I think as my focus is on immunotherapy, that's where I see the biggest breakthroughs, and really what I hope for and anticipate in the next 10 years is for us to be able to duplicate the successes that they've had in the leukemia, lymphoma, melanoma with CAR T-cell therapy in the solid tumors, not just brain, but also pancreas, prostate, lung, and I think we're on the verge of that. And I really do expect the next 10 years to see a whole new field for solid tumor immunotherapy.
Thank you. Yes, and you lit the world on fire with your papers over the last few months. It was on all the media outlets. Thank you for mentioning the Abramson Cancer Center. And we look for the next steps. I'll turn it over to Bo Ebby, who's our chair of the Breakthrough Challenge, for comments. Let's see if I can find Bo.
Great. You're always a tough act to follow. Remind me next year that I'd like to go first, so I don't have to pale in comparison. But thank you very much for another enlightening and impressive update. It's really tremendous the amount of work you do, and we're delighted to see how you maximize our dollars. And the 9-to-1 ratio is just a huge bang for the buck. And you're bringing hope to so many people. So we thank you and everybody that presented today. We're delighted that you're able to take the money and turn it into good work. On behalf of the board, I also thank all the participants and Chuck Zent and his family for the instrumental support they provide. As Bob mentioned, this year's ride is September 15th, and we look forward to catching up and rubbing elbows with the leading researchers and learning more about the latest advancements in cancer research. We are also planning a cocktail party this year for October 23rd that will be in South Philly at the Quartermaster to reflect back on everything we've accomplished this year. We are heavily into our fundraising. We're over $60,000 to date, well on our way to our $300,000 goal for this year. And if we accomplish that, we'll be close to $3 million lifetime. And again, we're delighted to see that the money is so well spent. So thank you all for joining, and we look forward to seeing you on September 15th.
Okay, and we'll leave it at that. Thanks, everyone, and thanks, Bo. And September 15th is the next hot ticket. Have a great day, everybody.